Environmental Effects on Embryonic Development by Danielle Ferrero and Sophia Gotzak. Abstract. This experiment was performed to observe and discover the environmental effects and optimum temperature at which chicken eggs would hatch. First, the eggs were prepared in an incubator for 56 hours in between the temperatures 34.5 degrees Celsius and 39.5 degrees Celsius. Then a cup was prepared to hold the unshelled eggs, which required a hole to be cut at the bottom of the cup. Saran wrap was to be used to secure the egg, along with water and calcium. Once the egg was successfully transferred into the cup, it is put back into the incubator, and then observed. The methods for this experiment were conducted in reference to an article that conducted a similar experiment. Also, for the results, a video was recorded showing the growth of a chick in the incubator. The significance of this experiment is with the conclusion one will know the optimal temperature for hatching chicken eggs.
Introduction. The overall purpose of this lab was to determine the optimal temperature for hatching chicken eggs. During the incubation of an egg, two key factors influence the development of the embryo. Those factors are humidity and temperature. For the specific lab, just temperature was explored and the humidity was kept at a constant 50%. When incubating chicken eggs, there is the lowest temperature the egg can develop in, which is 32 degrees Celsius, and the highest is 40 degrees Celsius. With incubating eggs, a hen, which is a female chicken, pulls out her feathers when she is ready to begin incubation. The hen removes her belly feathers so that her skin can provide the warmth the eggs need. The hen's body provides a temperature that is between 32 degrees Celsius and 40 degrees Celsius, and her sweat adds the proper humidity for the embryo to hatch. Hens are an efficient method of hatching eggs. However, with an incubator, many more eggs can be hatched at one time, and you do not require the hen to go broody. However, the incubator, unlike the hen, needs the temperature to be set. For the best hatch results, the question is, what is the most optimal temperature at which to hatch the egg? Over the different tests that were performed, a set of 5 eggs were incubated at each of the temperatures between the lowest and highest temperatures. To observe the development of the embryos, after the second day of incubation, the eggs would be removed from the shells and placed in an environment that resembled that of an eggshell.
Introduction continued. This method of watching the eggs outside of the egg was successfully executed by a high school class of students in Japan. During their experiment, they were able to watch the development of an embryo into the chick. Although this experiment was not the one we conducted, we used their method to observe the development of the embryo with different temperature changes. The embryo was cut from the shell after 56 hours and placed into a cup that was set back into the incubator. With the embryo being exposed in the cup, the embryo could be observed on a daily basis to view development. The experiment that was conducted was inspired by another experiment that went into more detail by actual scientists. The people who tested the experiment used more science and methods to determine how temperature affected embryo development, whereas this experiment used just mortality to determine what temperature was most optimal. The methods used in the experiment should be able to determine what the most efficient temperature to use when hatching chicken eggs.
Hypothesis, null and alternative. Null hypothesis. If the fertilized eggs are incubated at different temperature, then there will be no change in the development. Alternative hypothesis. If the temperature of the fertilized eggs increase from 32 degrees Celsius, then the development of the embryo will increase to an optimal point, where from that point it will begin to decrease until the temperature reaches 40 degrees Celsius.
Materials. The materials used in this experiment were a 250 to 300 milligrams of calcium lactate, half a teaspoon of distilled H2O, fertilized pre-incubated 55 to 56 hour eggs, distilled H2O, saran premium plastic wrap, high oxygen permeable, plastic food wrap with ventilation holes, cotton ball and hole for ventilation plus filter, a plastic cup, incubator, rubber band, and gloves.
Procedure. Step 1. A group of 5 fertilized eggs were collected and left at room temperature for 2 days. Step 2. The fertilized eggs were incubated in the shell for 56 hours at the temperature being tested, between 34.5 degrees Celsius and 39.5 degrees Celsius. Step 3. A cup was prepared for holding unshelled eggs. To prepare the cup, a hole was cut an inch from the bottom of the cup. Then a 10-inch by 10-inch sheet of saran wrap was placed over the top and the egg was pressed into it to mold the shape for an egg. A rubber band was placed around to secure the sheet. Water and calcium lactate were put into the saran wrap. The eggs were carefully removed from the shell and placed in the saran wrap base. Another 10-inch by 10-inch sheet of saran wrap with ventilation holes was placed over the top and secured with a rubber band. Step 4. The cup with the eggs was placed back into the incubator. Step 5. The eggs were watched and photos were taken to record development.
Data. The table shows how many days the embryos were able to survive under each subject temperature in the incubator. When all of the embryos for the temperature died, A new set of eggs were set on a higher temperature and recorded again for how many days they survived. During each temperature, a total of 8 eggs were incubated. The first 56 hours, the embryos will grow inside their shell. The days marked with an X had no surviving eggs past the 56 hours.
Data. The graph shows the highest amount of days the embryo survived under a certain environmental temperature.
Data. Embryo development video. The video is a compilation of the videos taken daily of the embryo until it reached mortality at day 16. The embryo was incubated on a temperature of 37.5 degrees Celsius.
Data. Embryo picture collage with days. The chart depicts a collage of the photos taken daily up until the embryo's death at day 16. The photos were taken approximately 6:30 each day. The embryo died at 12:30 p.m., therefore there was no picture of the embryo still living from that day. Day 1 and 2 were conducted inside the egg, therefore there is not a picture for either of those days.
Sources of error. There were multiple sources of error present during the experiment. One possible source of error is exposure to certain chemicals and bacteria during the second step of the methods. If the egg was not quickly transferred into the saran wrap, it could possibly be exposed. Also, sterile gloves had to be worn when dealing with the egg in general, so it could not be contaminated. Even though gloves were worn, there is still a chance for contamination of the egg. Another possible source of error is a little over halfway through the experiment, a power outage occurred, which caused the incubator to shut down. Although power did return about 45 minutes later, the embryo only managed to survive for a few days longer. It could have negatively affected it and caused it to die sooner than expected.
Conclusion. This experiment was conducted to determine the optimum temperature at which to hatch chicken eggs. The null hypothesis, if fertilized eggs are incubated at different temperatures, then there will be no change in the development, was rejected by the data collected from the experiment. It was shown that after the fertilized eggs were put into the incubator a second time at different temperatures, the growth and survival rate of the fertilized eggs did change in mostly positive ways. With each new subject, the fertilized egg, a different temperature that was increasing each time was used, and the changes became more prevalent with the increasing temperature, so that disproves the null hypothesis. The alternate hypothesis, if the temperature of the fertilized eggs increases from 32 degrees Celsius, then the development of the embryo will increase to an optimal point where from that point it will begin to decrease until the temperature reaches 40 degrees Celsius, was supported by the data collected from the experiment. As the temperature was closer to the middle, around 36.5 degrees Celsius and 37.5 degrees Celsius, the embryos were able to survive further into incubation. When closer to the two outer temperatures, the embryos did not make it much past the second day, especially with the colder temperatures. The lower temperatures proved to cause little development within the egg, while the higher temperatures, even though they were not as successful as the medium temperatures, still made it almost a week into incubation. The experiment conducted determined that the optimal temperature to hatch a chick are around 36.5 degrees Celsius and 37.5 degrees Celsius, and although the other temperatures are still effective in hatching chicks, they are not as successful or efficient.
Works cited. This experiment has been conducted before. However, it went into more depth. One experiment is from the Japan Poultry Science Association. Another experiment that influenced our own experiment was from the BMC Veterinary Research.